Do any of you like to garden? Or watch the lawn come alive with grass and flowers as the weather warms up? I sure do, and none of it would be possible without plants. My name's Valerie, and I'm the Outreach Coordinator for Future Energy Systems, or FES. FES is a research program at the University of Alberta focused on studying the energy transition. Thanks so much for joining us to explore plants. In this series of clips, you'll learn about the importance of plants, how to test if seeds grow well, and whether we can grow plants without soil. When you think of plants, what do you see? Giant trees? Pretty flowers? In this part, we are going to learn about the early life of a plant, when it changes from a seed to a seedling, which is called germination, as well as many of the factors that can affect it. You'll also learn how to calculate your germination rate. Here are the supplies you'll need. Get growing! I want you to all close your eyes and picture a healthy earth. What do you see? Animals? Clean water? The shining sun? All of those things are part of a healthy earth, but you know what I see? I see plants, all different types and sizes of plants. Why do we need plants? Well, there's lots of reasons. We all like to breathe, right? Plants produce oxygen through a process called photosynthesis, where carbon dioxide and water are transformed using sunlight. By transforming carbon dioxide into oxygen and storing carbon in their tissues, plants help reduce the effects of climate change because carbon dioxide is a key greenhouse gas. All animals, including us humans, require plants for food and shelter. And plants are essential for maintaining a healthy soil. Plant roots keep soil from eroding, and plant litter adds essential nutrients and other components as the plant breaks down into the soil. I could keep going on forever with all the great things plants can do, but I'll stop for now. But we remove plants when we build cities, cut down trees for building materials, and mine for important mineral resources. That's a problem because we know plants are important for a healthy earth, so we need to revegetate or bring back plants after these activities are done. Revegetation is a key step in land reclamation, which is the process of converting disturbed or damaged land back to what it was before or to something new. When we're revegetating an area, we have to decide what plants we want there. The decision may be based on what was there before, what the animals in that area need, what plants may be useful, or all of the above and more. Plants come in different shapes and sizes. Many plants have different growing or germination requirements. What is germination? Germination is when a plant emerges from a seed and turns into a seedling. Testing germination is an important part of revegetation and what we're going to do today. Any ideas why it's important? Well, let's think about it. If I want 10 plants to grow in an area, maybe I'll plant 10 seeds. If all 10 of those seeds germinate, that's awesome. That means we have 100% germination and that we'll have 10 seedlings, although they could die or get eaten later. But if only 5 of my 10 seeds germinate, that means I only have a germination rate of 50%. A lot of the time, many seeds do not germinate, and this could be because we don't have the right conditions. Maybe not the right lighting, or the right amount of water, or the right temperature for the seed to germinate. Knowing what the seeds need to germinate is really important when you're planting. It's also important to know how many of the seeds are actually going to turn into seedlings. Knowing germination rate means that we can plant the right number of seeds to get the number of plants you want in the end. So, we're going to test germination rate. How do we do that? We need to do some science. First thing we need are seeds. Seeds come in all different shapes and sizes. We're going to test the germination rate of 2 different species today, barley and blue flax. You can try seeds from your garden, from fruit that you eat like apples, or buy some from a store. So we're going to need some supplies. You need a container to put your seeds in. So I have a Petri dish, but you could also use a plate in a Ziploc bag. Then you need some paper towels. And finally, you need something with water, so a cup of water or a squirt bottle. So step one, you're going to take your container, your plate, or your dish, and you're going to put a folded up piece of paper towel in the bottom. Then you're going to squirt some water on it. You want to make sure the paper towel gets really wet, but you don't want any extra water in the container because that could mean that the seeds are floating. So let your paper towel get wet and then dump any extra water out into an extra container. Now if you're using a plate with a Ziploc, you would just put your paper towel here and then tuck your plate back in. Now we're going to add the seeds. I'm going to add 10 seeds because that gives us a large sample size and makes the math really easy. So you can add however many you want, but make sure you know exactly how many you added. Now, we need to put these two containers somewhere warm, but not too hot. And one thing to remember, if you're using a Ziploc and the plate method, make sure you don't seal the Ziploc because the seeds still need some oxygen. I'm going to put mine by the window where it gets some sun and is a bit warmer. It will take a few days to see if your seeds germinate. For a seed to germinate, it needs the right conditions. That could mean the right temperature, amount of water, or light. As I said, many different species have different requirements. Some seeds will germinate in the light, whereas others need the dark. Imagine if your seed was growing underneath the soil or was buried underneath a snowbank. It would need to be able to germinate in the dark. So maybe you should test your seeds in the light and in the dark. It may be too warm or cold where you're testing your seeds. Generally between 15 to 20 degrees Celsius is a good temperature because it's the temperature that we have in springtime when most of our seeds are germinating. So try your seeds in places all over your house that might have different temperatures and see if that impacts your results. You also need to make sure that your seeds stay damp. So anytime it looks like your paper towel might be drying out, add a bit more water. This can be really hard in nature because sometimes it might not rain for a really long time, or the soil where your plant is growing might not hold very much water. Some plants may never germinate. This could mean that it's dead or that it's dormant. That means that the seed is going through a sleep period that keeps it from germinating until the exact perfect conditions are met. We can break dormancy or wake the seed up by mimicking some of those conditions. For example, we could keep the seed somewhere really cold, which is like winter conditions that some seeds need to go through before they germinate. Other seeds might need to be eaten by an animal so that the hard outer layer of the seed can be removed. In this case, we can scratch the surface of the seed to encourage germination. Some seeds need to be exposed to different chemicals, temperatures, light, or even fire. When these environmental conditions are met, it tells the seed that it's the perfect time to grow, so the seed wakes up and germinates. For many species, we know what they need to germinate, but for others, we're still learning. So it's been a couple days. Let's see if we have any germination. We do. Do you see that little white part? That is the radicle which forms the root, the first thing that emerges from the seed. In nature, it will attach to the ground, keeping the plant where it is and starting to absorb water. So what happened during germination? First, the seed absorbs water and the outside starts to swell and soften. This step is called imbibition. Next is a lag period where things start to happen inside. The seed breathes and starts to make and store food and proteins. Finally, we have the step we see here, root emergence, where the plant starts to grow and emerge from the seed. Now, are you all ready to start recording your data? Download the data sheet from below the video and we'll get started. This is your data sheet. On it, we're going to record how many seeds germinate. Every day, check your container and count how many seeds germinated and mark it on your data sheet. Then, remove those seeds. The reason we remove them is because we don't want to accidentally double count them later. You're going to keep monitoring for at least a week or two until no more seeds germinate. So I've been monitoring my seeds for a couple weeks, and now it's time to figure out the germination rate. How do we do that? Little bit of math. So step one is to count the total number of seeds that germinated from each of your species. So for barley, I had 9 seeds germinate. And I know that I originally planted 10. So that means my germination rate is 9 out of 10, or 90%. With blue flax, on the other hand, I had 0 seeds germinate, which means I have a 0% germination rate. The reason that blue flax didn't germinate could mean that the seeds are dead, or, like we talked about, they could be dormant and we would need to break that dormancy before they would germinate. So what did you guys get? Did your seeds germinate? What was your germination rate? Let us know on Facebook or our website, and thanks so much for learning about germination with me today. I hope that you can get out there and revegetate all the places that you love. Bye!
How did setting up go? It'll take a few weeks to see if your seeds germinate, so good luck! Now you've learned all about germination, let's learn more about how plants grow. We'll be joining the University of Alberta Permaculture Group to learn about greenhouse gases and aquaponics. In this part, you'll get to do an experiment yourself and build a do-it-yourself hydroponic system, which is growing plants without soil. Here are the supplies you'll need. Have fun!
Hi, welcome to the University of Alberta Permaculture Group presentation. We will be talking about greenhouse gases and aquaponics along with some fun at-home experiments and activities. So a little bit about our group. Our goal is to build carbon neutral systems which function to provide food. Carbon neutral means that the net carbon released is equal to the amount we input. It does not change the amount of carbon dioxide in the air. The way we're achieving this is building an aquaponics system. Aquaponics has the potential to use 90% less water when growing crops, and this is a substantial amount since water is a huge part of modern day agriculture. Additionally, we have other projects such as our algae canopy, which will function to remove CO2 from the air, providing shade for aquatic life, or in urban environments as architectural features with positive environmental impact. So let's talk about the greenhouse effect. The greenhouse effect is basically sunlight is absorbed by the earth and some is reflected as infrared rays. The rays are trapped by greenhouse gases, which can absorb these infrared waves and radiate them or reflect them back towards the surface. More greenhouse gas in the atmosphere causes an increased retention of heat, which causes global warming, increasing the temperature of the Earth's surface beyond normal levels. Greenhouse gases include, but are not limited to, carbon dioxide, water vapor, and methane. We can actually test this with a simple experiment. In an experiment, you want to keep everything the same except for one thing. That is how we know that the thing we changed actually caused the effect we saw. In our experiment, you're going to need a few things. 2 similar clear containers, anything that's around the same size. An elastic, 2 ice cubes roughly of equal size, some cling wrap, damp soil, and a sunny windowsill or a bright light bulb. So how we're going to set up this experiment to prove the greenhouse gas effect is to place an equal amount of damp soil in both containers. Then place an ice cube on top of them at the same time preferably, and then cover one of the containers with cling wrap and secure it with an elastic. Then put both containers in a sunny window or the same distance from a bright light bulb. Let's start with some questions. So, which ice cube melted faster? Or which one do you think will melt faster? Would it be the one with the cling wrap on top or the one open to the air? Also, what do you think about the cling wrap affecting the melting time? Does it make it shorter, faster, or make it stay the same? So, basically how this experiment works is the cling wrap acts like the greenhouse gases by absorbing some of the heat and radiating it back as well, as reflecting some of the infrared rays back. It's also clear like the air, so the ice cubes receive the same amount of light. So by doing this, it increases the relative temperature in that space between the ice cube and the cling wrap. This will in effect make the ice cube melt faster. So let's head on to aquaponics. Aquaponics is a system that combines aquaculture, which is raising of fish or other aquatic organisms, something like a salmon farm, with hydroponics, which is growing plants in water. And this is done in a symbiotic environment. In the diagram on the right, you can see that there's a relationship between the bacteria and the plants and the fish with the bacteria. How does this work? Well, looking at the aquaponics system on the bottom left, you can see here that if we start with the fish who are fed fish food and are growing, they produce waste. And this waste is pumped through a biofilter. This biofilter has bacteria in it. So the bacteria take that debris and fix the ammonia, the nitrogen in the ammonia, into nitrates. Now, nitrates are a very important part of plant fertilizer. So what we do is we take those nitrates and pump it into the tanks holding the plants on the top there. These plants use the fertilizer nitrate and grow, thereby taking out the nitrate that are bad for the bacteria as well as bad for the fish. And then this recycled water is given back to the fish clean. Now, basically, this is what a symbiotic relationship is. A symbiotic relationship is any relationship between organisms. There are three forms: mutualism, parasitism, and commensalism. Our system is a complex mutualistic relationship where the toxic excretions of the fish are removed by the bacteria, which use the ammonia and create nitrate as waste. These nitrates are then used by the plants to grow and then remove them from the system. All three organisms benefit. The plants get nutrients, nitrogen is the fertilizer, the bacteria fuel their metabolism, and the fish get clean water. The earth has finite resources which we need to use responsibly to care for ourselves and the earth by limiting resource consumption to a sustainable level. And this is how we increase that efficiency to allow more people to get more food. It harbors a lot of potential, especially in combating food scarcity. There are other benefits as well. If everyone can have this in their home, it means that we do not have to transport as much food to remote areas, and as well as people in remote areas can have food year-round should they have stuff like this. We can also use this technology to use real estate for farming that wouldn't naturally be used, such as rooftops or city skylines. So now we have a do-it-yourself hydroponic system for you. And it's not the same as aquaponics system because it does not have fish, but it is an easy at-home proof of concept of growing plants in water. Surprisingly, the plants will receive enough nutrients and will grow just fine. And there's a ton of plants that you can grow, and you only need a few things. So the most important one is a plant, preferably a young one with its roots still intact, because the roots are the plant's lifeline, and you want to be especially careful about that. A pop or a soda bottle, just used as a container and a plant holder, feel free to innovate or use what you have. It's just meant to repurpose something that you have, and as well as some water. What you're going to do is carefully remove a small plant from the soil, making sure to keep the roots intact. You can do this by making the soil wet or by just very carefully wiggling the plant out. If you pull the plant out without its roots, you will have to use another one. Next, you want to have an adult carefully cut a plastic bottle in half, similar to the one on the right of the screen there. Be especially careful, sometimes it can be a little sharp. Take the base side of that water bottle and fill it halfway with water. We will be flipping the other side with the mouthpiece into the water. You don't want to fill it all the way up. Halfway should be fine. Then you want to gently, very gently, pull the plant roots through the opening of the top half of the bottle. This will allow you to stabilize the plant as well as put it into the water in the next step. So when you're putting the roots in the water, you don't want the full root to be fully submerged. That way there can still be some gas exchange for the plant roots to breathe. Next, you want to place it in some sunlight, and if the water level drops, just add some more water to get the roots submerged again, but once again, be careful about overfilling. Now, there are tons of plants to use, and here are some cool ones. You can do some African violets, some fiddle leaf fig, some spider plant. There are so many, and we've even got more slides of them. And all you have to do is either take a cutting or anything like that, but the most important thing is if it's a small plant, you want to have it with the roots. So here are some more plants that you can actually eat. We got basil, chives, oregano, mint, rosemary, and sage. There are a lot more. Feel free to Google them. We thought these were some awesome ones that you can actually use right away, so feel free to try them out. This is a rendition of kind of what it would look like. You want the roots to be partially submerged in the water and the plant to be above the top of it, so then it gets enough sunlight and isn't hindered by anything dark or not transparent. As you can see, the thing with the water bottle is that it is transparent, so even though some of the light is blocked, the leaves will still get enough sunlight anyway. And you can feel free to mess around with this. Thank you for learning with us. We love doing things like this. Feel free to send us your results or tag us on social media, plus our email permagerp@ualberta.ca. Ask us any questions and any ideas you may have, as well as send us your video of your hydroponic system or what you thought of the greenhouse gas experiment, as well as check out the document below for the answers to the questions of today's lesson. Thank you.
Plants are all around us and play an important part in our lives. Thank you so much for joining us to explore plants today. Make sure to subscribe to Future Energy Systems YouTube channel so you don't miss any of our exciting content, and check out the links below. There's so much to learn as we explore our energy future.